Dr. Patricia Benito from Caldic Ibérica, and also she's working in the IBMCP in Valencia, Instituto de Biología Molecular y Celular de Plantas, and we're eager to hear her talk. The floor is yours.
Hello, good afternoon, everyone. I try to share the presentation. Okay. You can see it? Yes.
Yes.
Just one thing I got.
Okay, good afternoon. My name is Patricia Benito. Thank you for the organization for this opportunity to present the work rapid method to evaluate in a combinatorial manner of the synergistic effect of different plant biostimulants under abiotic stress. This work was carried out in collaboration between the Institute of Molecular and Cellular Biology of Plants and the company Caldic Ibérica. The anthroponic climate change is having an impact on the climate variability. The current observed of global warming trend has been 0.6 plus minus 0.2 degrees since 1900. It's lately continued to increase by between 1.4 and 5.8 degrees over the period 1990 to 2100. The high frequency of stress events, such as high temperature, salinity, and drought, can harm crop and reduce yield. One of the causes of the anthroponic climate change is the emission of greenhouse gases. The amount of the greenhouse gases emission from agriculture is significant, contributing between 11 and 21% of the global emissions. The most common emissions from agriculture practice are nitrous oxide, methane, and carbon oxide. The climate change, together with the inappropriate agriculture practice, are accelerating the soil desertification. This incrementing the effect of the abiotic stress in the agriculture crops. The most notable abiotic stress are the extreme temperatures, flood, drought, salinity, and heavy metals. In the case of the drought and salinity, are very important because in Europe, in the south of the Europe, such as Iberica Peninsula, suffer from a coexistence of salinous soils and a prolonged drought, which restricts the production of many crops. This is a very problem because this area of the Europe is our largest production of the fruits and vegetables. Due to the need to find an efficient solution to address the current agriculture problems, are operate the different products on the market, such as the biostimulants. The plant biostimulants and products of natural origin can improve the crop productivity under abiotic stress. In addition, these products are not considered agrochemical. For this reason, is common use in the sustainable agriculture. In addition, these products are made attention for its potential to mitigate the greenhouse gases emission in agriculture. The European Biostimulant Council, Industry Council defines biostimulant as the substances or microorganisms when we apply into the crops or resource field is to stimulate the natural process to benefit nutrient uptake and nutrient efficiency, improve productivity and quality of the crops, and for me, it's very important, is improved to tolerant to abiotic stress. However, there are few products on the market that combine different natural extracts or microbial and non-microbial biostimulants that synergistic enhance their effect against abiotic stress, such as salinity and drop. For this reason, the main objective of this work was to design a novel methodology to evaluate the effect of the combination of several natural extracts with biostimulant potential under abiotic stress. In the first phase of our project, we evaluate the biostimulant effect of 11 natural products. This product was from Caldic company. In this screening, we first performed in vitro tests using two model organisms, the yeast Saccharomyces cerevisiae and the plant Arabidopsis thaliana. First, we evaluate the biostimulant effect of our products individually or in combination with them on the stationary and continuous growth of Saccharomyces cerevisiae. After this, the biostimulant potential of our product was evaluated on seed germination and vegetative growth in Arabidopsis thaliana. The case of the combination of this product was made as follows. The biostimulant effect of the individual products are evaluated under normal conditions and in abiotic stress conditions. The natural compounds with a best biostimulant effect were combined with the rest of the products to produce the binary combination. Secondly, these combinatory combinations with the best synergistic effect were chosen to make a ternary combination. It means the binary combination was combined with the rest of the products, but was discarding these binary combinations with the negative effect. The combination was processed to repeat until they obtain the quaternary combination. But first of all, of this screening, we determinate the optimal dose of each natural process individually under normal conditions. To do this, we test different concentration of the biostimulant from the 2,000 to 20 milligrams per milliliter. In the most cases, the optimal dose was 500 milligrams per milliliter, except in the case of the products B9 and B10, that the show was lower. We first evaluate the effect of our products individually in stationary yeast grown by drop test under different abiotic stress, salt, osmotic, and temperature stress. We said that the product B3, B4, B5 showed the best results, especially under in presence of sodium chloride, lithium chloride, and sorbitol. I only during this presentation, I will only show the result of the individual products and the quaternary combination due to the complexity of this data. In this case, the individual screening, the product B9 and B10 have a deleterious effect in all stress tested. In the quaternary combination, the best combination was the product B2, B3, B4, B5, and the concentration of 100 milligrams per milliliter of each compound. Especially in presence of sodium chloride, lithium chloride, and sorbitol. In the case of sorbitol, is similar to the osmotic stress. However, the product B9 and B10 had antagonist effect in all combination, in the binary, ternary, and quaternary combination. Here, I want to show you an example of the effect of the quaternary combination B2, B3, B4, B5 on Saccharomyces cerevisiae grown under normal conditions, salt stress, osmotic stress, and temperature stress. Secondly, we make the we evaluate the effect of our products individually on continuous yeast growth. As we observed in this drop test, the products B3 and B5 was the one that present the best results in normal condition and in presence of lithium chloride and sorbitol. In the case of quaternary combination, again, the combination B2, B3, B4, B5 was stand out again and was the only combination with the biostimulant effect on continuous and stationary yeast growth. However, during all evaluation of the 11 products, we have observed that the compounds B9 and B10 showed a deleterious effect on yeast growth. These products are yucca extracts, and these extracts have a characterization is have saponins and polyphenolic compounds. These bioactive compounds has a strong antifungal effect. For the reason, in 2022, we published this paper where we describe the effect of yucca extract in seed germination of different plants and its antifungal power against yeast. Here you can see the aloe, the antibiosis aloe produced of the yucca extract. After the testing of the effect of our products in yeast, we evaluate their effect in plants using Arbona italiana. The evaluation of these products have the similar effect between yeast and the plants. The product that showed the best result was B2 and B5. You can see here in the chart the difference between the different products. In the case of the quaternary combination, again, the formulation B2, B3, B4, B5 at the concentration 50 milligrams per milliliter of each compound had the best results under normal conditions and present is a stress. However, in this case, this formulation did not show a biostimulant effect in plants growth under abiotic stress. Here, I saw the difference of the size of the plants between the control and the formulation B2, B3, B4, B5. The effect of the description of this method you can see you can find in this paper a fast method to evaluate in a combinatorial manner the synergistic effect on different biostimulants for promoting growth of tolerance against abiotic stress. Is similar the title of this presentation. For further testing, we consider the quaternary combination B2, B3, B4, B5 as Calbio. This is the name of the you can find in the market. Calbia is composed of yeast extract, Ascophyllum nodosum extract, licorice root extract, and white willow extract. After in vitro assays carried out in Saccharomyces cerevisiae and Arbona italiana, we want to analyze the effect of Calbio in crops of agronomic interest and sensitive of salinity, such as tomato, all lectures. For this, in the greenhouse access, the main objective was characterize at the physiological, biochemical, metabolic, hormonal, and transcriptional level the effect of Calbio combined with a microbial biostimulant under normal and salt stress conditions. And these greenhouse assays had for treatments control the product Calbio, the bacterial strain Bacillus BM08, and the combination of Calbio with the bacteria BM08 under two conditions: no stress and salt stress. In this assay, we observed that the combination Calbio with BM08 was the treatment with the highest production, especially under salt stress. Here, the percentage is a comparison of Calbio with BM08 with respect to the control. You can see the difference is great. Here, I want to show you the difference of the size of the plants. It depends on the treatment. Here in tomato, you can see we can compare the plants with Calbio BM08 with the control. Here the same in lectures. Among all parameters measured in roots and leaves, the analysis of metabolites stand out. We observed that the combination of Calbio with BM08 promote the production of isopentenyl adenosine in both tissues and in both plant species under salt stress, but this phenomenon not observed under normal conditions. The isopentenyl adenosine is a cytokinin. For this reason, we analyze different pathways or relationship with cytokinins, and we observe that the transcriptome analysis to lectures tissues treated with Calbio and BM08 under salt stress stimulate or show a larger number of unregulated genes related to synthesis of cytokinins. The result obtained in the lectures work you can find in this paper. The combination of microbial and non-microbial biostimulant increased yield in lectures and the salt stress condition by upregulating cytokinin biosynthesis. For the conclude, these results show that the use of Saccharomyces cerevisiae and Arbis italiana is an excellent system for the identification and characterization of biostimulants, and moreover provides an excellent platform to evaluate the synergies between them. The combination B2, B3, B4, B5 show a great biostimulant effect under salt and abiotic stress condition in yeast and salt stress in Arabidopsis. The greenhouse assays show that Calbio is the quaternary combination B2, B3, B4, B5 has a biostimulant effect alone, but especially in combination with the microbial product Bacillus BM08 under salt stress condition in lectures and tomato plants. And the Calbio with BM08 treatment stimulates the production of isopentenyl adenosine hormone related to the response to abiotic stress. This is our group. Thank you for my boss, Jose Miguel Molet, for the opportunity to participate in this project, and the group Abiotic Stress, Potassium Transform in Abiotic Stress in Plant and Yeast for all help in this long process. This project was funded by the CDT and the State Research Agency. Thank you so much.
Thank you very much. I was looking for the microphone. It got it was not lost, but I couldn't find it. So thank you very much for your thorough presentation. I think it's very interesting. And we'll give the opportunity to ask some questions. We have questions online. Not yet. So I have a question.
I have a question. Thank you so much for your nice talk. I was wondering if this type of biostimulants, the treatments and the protocols that you have selected so far, can be helpful also to protect against, for example, heat stress, besides the salt stress. Do you have any idea or any data?
I don't know, but we test this product, Calbio, and they drop the stress, but in broccoli.
Okay.
The result was wonderful. We don't know.
One minute. Could you please silence your microphone? We have a lot of feedback here.
Okay.
Thank you.
We don't know which, when we exposed Calbio, or the quaternary combination B2, B3, B4, B5 in Arabidopsis, we had these results. We don't know why the response is bad, but in the case of other plants, is perfect. In a greenhouse or in a field, Calbio have great results under drop conditions.
Thank you very much. I have a question as well. So the system you described it's an important breakthrough because testing different combinations of biostimulants or even priming agents that could have opposing effects on the plant material. I think that needs to be tested beforehand you move all through the entire experimental development. So what would you think, that's maybe I'm asking too much, maybe I'm asking a personal opinion, what would you think will be the next step in this quest for new effective biostimulants?
Okay, with respect to, in the case of Calbio, but in the rest of the biostimulant, the problem is the disponibility of novel products in the market. And now the people use the typical ascophyllonodosum, yeast extract, but alone. In the future, is your question, no? Probably the combination of microbial and non-microbial biostimulant is the best option, because you combine it, okay, in the case of the no-microbial biostimulant, you apport nitrogen or surfactants, but in the case of microbial biostimulants, you can have a relationship between plant and bacteria, and you can reach not only the plant tissues, you can reach the soil. This is very important. Not produce this year, you need produce during 10 or 20 years. Prolong this effect during a long period.
That the next step will be just to have like auto protective crops to maintain production at a steady level.
No think, okay, and this year I need to produce, I don't know, 50 tons. No, you need produce during 10 years, I don't know, 10 tons per hectare.
We had a question in the comment box. We don't have any questions. Thank you very much.
Thank you.
Very nice talk. So we move to the next talk. We won't stop. We decided not to stop this section. So please, Alma, could you introduce next speaker?
Yes. Yes, please. Yes, thank you. So I'm very happy to introduce Wanda Waterworth. Her seminal work on the DNA damage response in seeds is a relevant reference point for all the people working on these issues. So thank you so much, Wanda, for being here, and the floor is yours, please. Thank you.
Okay, well, firstly, thank you very much for the invitation to speak. I'm just figuring out how to link my presentation. So I click on the Present Now button.
You have an icon with a square and an arrow, upwards arrow. Yes, you got it. Perfect.
Okay, can you hear me and see the screen now?
Yes.
Okay, so everything's ready to go. Okay, so at the moment I can only see my slides, so I'll just go through my lecture. Okay, so today I'd like to present my recent work investigating the importance of safeguarding the genome in germination and seed longevity. So the desiccation-tolerant seed represents a highly successful survival strategy, and this is beautifully illustrated by these date palm seeds from Israel, which were capable of germination after 2,000 years. However, seeds deteriorate with time, and exposure to adverse environmental and storage conditions. A decrease in seed vigor and germination performance is first manifest as a progressively increasing lag period to the completion of germination. This decrease in rapidity and uniformity of germination, known as loss of germination vigor, eventually culminates in the loss of seed viability. Now, understanding seed germination performance and seed longevity is very important to sustainable agriculture, because germination performance is an important determinant of seedling emergence and final crop yields. It is also very important to plant survival in natural ecosystems. The panels on the right illustrate the impact on seedling emergence using low and high vigor rice seedlots respectively. However, to date, our molecular understanding of seed vigor remains incomplete. For a number of decades, it's been known that as seeds age or deteriorate, they accumulate damage to biological macromolecules such as RNA and proteins, and also membranes and organelles. My work focuses on the link between seed aging and damage to the genome. Of all the biological macromolecules, DNA is irreplaceable. It represents both the genetic material of inheritance, but it is also the template for gene expression and DNA replication. However, DNA is inherently unstable in the cellular environment, and in fact, during the course of the next hour, every cell in your bodies will accumulate 6,000 damaged products. In the plant cell, the genome is constantly under assault from environmental stresses such as ultraviolet radiation and heavy metal contaminants in soils. However, a significant amount of DNA damage arises from the byproducts of cellular metabolism, in particular reactive oxygen species. If unrepaired, DNA damaged products inhibit transcription, DNA replication, and cause permanent mutations in the genome. So if unrepaired, the plant exposed to DNA damage will carry developmental defects, growth inhibition, and this can eventually result in cell death. The seed stage of the plant life cycle is associated with particularly high levels of genome stress. Whilst on the mother plant, the developing seed is exposed to adverse environmental stresses, which is a problem which is likely to become more pronounced with rapidly changing climates. Desiccation and rehydration themselves are associated with enormous amounts of genomic stress. And in dry quiescent state, seeds accumulate damage in the absence of active repair mechanisms. And it's been known for a number of decades, in particular from the work of Daphne Osborne's lab, that DNA repair is one of the first events that occurs upon seed imbibition, and this repair precedes the onset of the cell cycle by several hours. As a consequence of the lethal consequences of DNA damage, organisms have evolved sophisticated DNA damage response and repair pathways. The double helix structure can contain a range of DNA damaged lesions, and these may vary in severity from oxidative base damage through to single and double strand breaks. The double strand break, representing the complete severance of a chromosome, is highly mutagenic and highly cytotoxic. To put things into context, a single double strand break can kill a yeast cell if unrepaired. However, higher plants, as we will see, have evolved a higher tolerance. In general, the specific DNA damage lesions are repaired by specific multi-step repair pathways. Now, a number of years ago, we identified that Arabidopsis mutants, defective in repair of double-strand breaks, are hypersensitive to seed aging. In this slide, we're looking at the germination of mutants deficient in the DNA ligases 6 and DNA ligase 4. We're looking at their germination after 8 days after accelerated aging. We see that the germination of the DNA ligase deficient seed is reduced in comparison to the wild type, and the germination of the double 6-4 mutant is even more greatly affected. So this work established the genetic link between double-strand break repair and seed longevity. In more recent work, we've used natural aging of Arabidopsis DNA ligase seed lots to check that the results we see with accelerated aging are similar in natural aging. And you can see by the dashed lines that we see reduced germination vigor of the DNA ligase mutants as compared to wild type. So this data was obtained with a 10-year-old seed lot of the wild type and the DNA ligase lines. And so it's nice to know that the work that I'm going to present with our DNA repair deficient mutants is similar in both accelerated aging and natural aging. So we've seen that there are a number of different repair pathways for different DNA damage lesions. So do all of these have roles in seed longevity? Well, work from our lab and a number of others has established that all the major repair pathways in plants do have important roles in seed longevity. Recently, we used comparative genetic analysis using mutants defective in each of the major repair pathways found in plants, summarized here. And we compared the germination performance of each of these mutant lines with wild type. As shown here, we see little difference between unaged DNA repair mutant lines and wild type. But as aging progresses, we see that germination vigor is decreased in all DNA repair mutant lines. This slide summarizes the final viability of the unaged and the aged lines, and we conclude that all repair pathways impact on seed longevity, but that the most obvious phenotype was seen with mutants deficient in double-strand break repair. So here we see the greatest loss of viability after aging in mutant lines deficient in XRCC2 and Q70. So both of these mutants are deficient in repair of double-strand break repair, but in different pathways. XRCC2 functions in the homologous recombination pathway. That is a pathway in which the broken DNA ends are mended as they were originally found. However, in plant cells, a pathway known as non-homologous end joining predominates. So the Q70, Q80, and DNA ligase mutants that I've presented so far all participate in the non-homologous end joining pathway, which joins broken DNA ends together rapidly but randomly. So we've established that double-strand breaks are rate limiting for germination because mutants deficient in double-strand break repair show hypersensitivity to seed aging. So how can the presence of a chromosomal break influence germination? A number of years ago, we hypothesized that the delayed germination of aged seeds could be mediated by DNA damage signaling. So how do cells signal DNA damage? Well, in response to detection of a DNA damage product, cells or eukaryotic cells signal DNA damage through the master kinases ATM and ATR. These represent the major sensor kinases, and ATM generally responds to double-strand breaks, whilst ATR largely responds to replication stress. These kinases orchestrate and coordinate the DNA damage response. These kinases have hundreds of downstream phosphorylation targets, and phosphorylation cascades activate a number of downstream responses in the DNA damage response. They activate checkpoints which slow or halt progression of the cell cycle. This gives the cell more time to repair DNA damage. We also see the induction of the transcriptional response to DNA damage, which is a transcriptional response enriched in genes important for DNA repair. We see the activation of DNA repair signaling and repair proteins. And finally, in the presence of unrepaired damage, we can see the activation of pathways to program cell death. So what is the germination phenotype of the mutants deficient in ATR or ATM? So ATR and ATM represent the major kinases which control the plant DNA damage response. We identify little difference between wild type and mutant lines in unaged high-quality seed lots. However, as aging progresses, we start to see that mutants deficient in ATM, as shown by the red line here, we see that these mutant seeds germinate faster than their wild type and ATR mutant counterparts. So after 7 days aging under these conditions, we see a characteristic delay to germination seen as seed vigor is lost. However, the ATM line appears to lack this lag period to completion of germination. And as aging progresses, we see that ATM and ATR mutant seedlots demonstrate a higher viability than their wild type counterparts. So these seeds are deficient in the checkpoints which control the cell response to DNA damage. We conclude that ATM regulates germination progression, that is seed vigor, but both ATM and ATR influence the viability of aged seeds. So these seeds are germinating in the presence of apparently large amounts of DNA damage. So in order to confirm this, we looked at the levels of chromosomal abnormalities in germinating wild-type and mutant seeds. So on the right, we can see a typical chromosomal abnormality. This is termed an anaphase bridge, and it occurs when a broken chromosome has been misjoined by the host cell's double-strand break repair machinery. And these are very cytotoxic lesions. However, we identified that even high-quality seed lots of both wild-type and mutant seeds germinate with 1 to 2% of their cells carrying chromosomal abnormalities. This would not be observed at any other stage of the plant life cycle. And furthermore, when we look at seeds which we have aged for 7 days, as I previously showed you, these seeds have lost germination vigor but not viability. We see that wild-type seeds are able to germinate with 4% of their cells carrying chromosomal abnormalities, and this increases to 8% in ATM mutants, which are deficient in the DNA damage checkpoint. So this is remarkable because most living organisms cannot tolerate this level of DNA damage. And in fact, this is very similar to the previous observations of the lab of Eric Roberts back in the 1960s and 70s. He identified the occurrence of chromosomal abnormalities in seed aging of a number of crop species, and he concluded that there is no threshold level of seed viability before chromosomal aberrations occurs. So these mutant seeds are apparently germinating with a higher seedlot viability than wild type. However, when we look at the development of seedlings germinated from the ATM and ATR seedlings, we identify that although the seedlot viability is higher, the seedlings show reduced survival on soil, and as shown here, the development of true leaves is significantly delayed in seedlings germinated from aged ATM and ATR seeds. So how does ATM control the delayed germination in aging seeds? We hypothesize that it could be through control of the cell cycle. So this slide summarizes the cell cycle in germination. In the germinating Arabidopsis seed, most cells pass most of germination in the resting or G1 phase of germination and only pass into the first phase of cell cycle, that is DNA replication or S phase, towards the end of germination. You can visualize this by looking at the incorporation of EDU into replicating nuclei. So on this slide, all nuclei can be visualized as purple nuclei using DAPI staining. The appearance or the stage of germination is indicated at the bottom of each panel, and we can see that at the point at which the Arabidopsis seed coat is rupturing, we start to see the appearance of S-phase nuclei, and this is EDU labeling of newly synthesized DNA as the cell cycle begins initiation in the root apical meristem. And as we see the progression to completion of germination with the emergence of the radicle through the seed coat, we can see that most of the cells in the meristem begin to progress into S phase. So we ask the question, is the timing of cell cycle different in ATM mutant seeds than wild type? So just to remind you, we see no difference in the speed of germination in unaged seed lots, but as aging progresses, we see that the ATM mutant line germinates significantly before wild type and ATR. So we looked at the timing of cell cycle initiation by counting the number of embryos containing the EDU labeled green nuclei, and graphically presented on the left, we can see that shown by the dashed lines, wild type and mutant lines show a similar timing of DNA replication in unaged high quality seed lots. However, as aging progresses, using the seed lots that we saw on the previous slide, we can see that ATM mutant seeds display the initiation of S phase over 24 hours before that of wild type and ATR mutant seeds. So this work demonstrated that ATR is regulating germination and the induction of the oligophace to germination through control of cell cycle activation. And we also showed that this is achieved by transcriptional inhibition of the cell cycle inhibitors ASMR5 and 7. So we've shown that seeds can withstand remarkably high levels of DNA damage. So how do seeds mitigate the effects of high levels of genome stress on germination and plant growth? Well, we hypothesized that seeds could display a higher resistance to DNA damage than plants at other stages of the plant life cycle. So to test these hypotheses, we did a very simple experiment in which we took an early imbibing seed. So actually, these seeds have been stratified at 4 degrees for 48 hours, and then we transfer them to room temperature. And we compared the root growth from the early imbibing seed with that of the 7-day-old Arabidopsis seedling after exposure to 100 gray dose of X-rays. So essentially, in the next series of experiments, we're going to take the seed and the seedling, we're going to expose these to 100 gray dose of X-rays, and then we're going to measure root growth in the following days. We found that when we look at the root growth post-irradiation, that in the 7-day-old seedling, we see a substantial growth arrest post-irradiation in the irradiated seedling relative to the control. However, when we look at the root growth, which occurs subsequent to irradiation of the seed, we see little difference in the subsequent root growth. So we conclude that seeds and seedlings display very different responses and resistance to DNA damage. And when we look at this loss of resistance to DNA damage in more detail, in this slide, we have taken seeds at different stages of germination, as for example, seen on the bottom panel. We take seeds at progressive stages in germination, expose them to 100 gray dose of X-rays, and then look at the root growth, which is presented in this graph as millimeters per day. We see that there is little difference in the mean root growth post irradiation. In the control seeds which have not been exposed to radiation, we see little difference in final root growth as the seeds progress through germination. However, as we irradiate seeds progressively through advancement of germination, the subsequent root growth shows increasing sensitivity to DNA damage. So in other words, the loss of resistance to DNA damage occurs progressively And this may be comparable with the loss of desiccation tolerance that we see in seeds as they advance through germination. So we next hypothesized that the difference in response to DNA damage that we see between seeds and seedlings is underpinned by developmentally different responses to DNA damage. A hallmark of the DNA damage response is programmed cell death. We can visualize programmed cell death by using prapidium iodide staining, which stains dead cells red. And the Arabidopsis seedling root response to DNA damage is well characterized and presented here. So here we have a Arabidopsis seedling root. When we irradiate roots, we see the induction of programmed cell death in the actively dividing cells of the root meristem. So we can see here PCD as the red cells. We can also visualize the location of the quiescent center. That is a pool of slowly dividing stem cells by using the WOX5 DFP reporter. So in seedlings and mature plants, a response to DNA damage is the induction of PCD in the root, and this represents a highly effective mechanism to eliminate cells with compromised genomes, as this pool of cells represent the progenitors of future cells in the plant. So do we see a difference in PCD between seeds and seedlings? So as before, we expose our early imbibing seed and our 100 and our Arabidopsis seedling. We expose both these to 100 gray dose of X-rays, and then we look at the progression of programmed cell death over the next 4 days. Looking on the top panel, we can see that in the Arabidopsis seedling, after irradiation, we start to see the incidence of large amounts of programmed cell death from 16 hours post-irradiation, and that this progressively increases during the next few days and is accompanied by extensive meristem disruption. So what happens after irradiation in the seed? Well, actually, we see very little incidence of PCD in the root produced post-irradiation. And interestingly, we only start to see the appearance of a little bit of PCD around 3 days post-irradiation. Looking at this in more detail, we can see that the seedling response to DNA damage is a over 2-fold expansion of the root and the root apical meristem, and we see very little change in the irradiated seed. So we conclude that the seed and the seedling have very different developmental responses to DNA damage. So is this reflected in the transcriptional response of seeds and seedlings to DNA damage? So the transcriptional DNA damage response is under control of ATM, and it includes the induction of hundreds of genes enriched in functions such as DNA repair and chromatin remodeling. We previously identified, as shown here by expression of the marker gene RAD51, we previously found that the plant DNA damage response is induced within 3 hours of imbibition in Arabidopsis. We see a large DNA damage response here shown by the marker genes RAD51 and PARP2, even in germination of high quality Arabidopsis seeds. And this is larger than at any other point in the plant life cycle. We also see the induction of the transcriptional DNA damage response, again using the RAD51 51 marker gene here, we see the induction of the DNA damage response in germinating barley seeds is shown here. The time from imbibition shown on the bottom axis. However, when we take barley seeds which have not undergone maturation drying on the mother plant but are capable of germination, we see little induction of DNA damage response. We therefore conclude that in the germinating seed, the DNA damage response is a direct consequence of desiccation, quiescence, and rehydration. So do we see differences in the plant DNA damage response between seeds and seedlings? So to answer this question, we again irradiated the early imbibing seed and the seedling and isolated RNA from each after 6 hours. We then looked at the genes which are responsive to DNA damage using RNA-seq analyses. This summarizes our findings. In the pink circle identifies the genes which are upregulated in response to X-ray in seeds, whilst the orange circle represents the genes upregulated in seedlings in response to X-ray. We identify that a large number of these genes are similar and that we see similar expression of core DNA repair genes in seeds and seedlings. However, when we look at the genes which decrease in expression in seedlings, as shown by the large blue circle, and the genes which are downregulated, as shown by the small purple circle, we see that there is significant difference between the DNA damage responsive genes. So we identify in particular that cell cycle associated genes are downregulated in the irradiated seedling, but not the seed. So this identifies that there are significant differences in the transcriptional DNA damage response between seeds and seedlings. So to summarize this part of my talk, how do seeds mitigate the effects of high genome stress on germination and growth? Well, we conclude that distinct physiological and transcriptional DDRs in seeds and seedlings promote germination and subsequent growth. And in germination, low cell cycle activity and DNA repair mechanisms mitigate damage incurred in the seed. And as we've seen, programmed cell death and meristem disruption occur with very low frequency before germination. And we hypothesize that in the early growth stages, rapid primary growth is prioritized over meristem maintenance. In other words, we propose that it is absolutely essential for the germinating radical to establish itself in the soil, to provide the growing seedling with nutrients and water. So in this part of my talk, we've looked at the plant response to X-rays, a situation which the plant is not likely to encounter in the natural world. So do we see a similar response to DNA damage occurring in seed aging? So to look at this in more detail, we're going to use seeds deficient in the transcription factor SOG1. So SOG1 is a transcription factor which plays a very important role in the plant DNA damage response. It integrates signaling from both ATM and ATR. And it is unique to plants, but it acts in a manner analogous to P53 in mammalian cells, in that it controls programmed cell death. So SOG1 controls many of the downstream responses of the plant cell to DNA damage. So what's the germination phenotype of seeds deficient in SOG1? As we previously saw with ATM and ATR, we see little difference in unaged seed lots. However, as aging progresses, we can see that the SOG1 mutant lines show a higher viability than their wild-type counterparts. Here, the SOG1 seed lots have around a 40% viability after 14 days aging, as opposed to the 20% displayed by the wild-type. So SOG1 mutant seeds have a similar phenotype to ATM and ATR, which also control the plant's DNA damage response. However, as we previously saw with ATM, we predict that the lack of DNA damage response mechanisms means that these seeds are carrying a high level of DNA damage. When we look at the survival on soil of seedlings germinated from aged seeds, we can see that the survival of the SOG1 mutant seedlings is lower than that of the wild type. In other words, these seeds show a apparently higher seed viability, but the DNA damage is impacting on seedling growth. So what does SOG1 do? Well, I previously showed you the well-characterized response of Arabidopsis seedling roots to DNA stress. So again, looking at Arabidopsis seedling roots, just to recap, we see no PCD or little PCD in Arabidopsis roots of either wild-type or SOG1 mutants grown under non-stress conditions. However, when grown on a chemical bleomycin which induces double-strand breaks, we again see the induction of cell death in the actively dividing root meristem cells in the wild type. However, this is absent in the SOG1 mutant roots, indicating that in response to DNA damage, SOG1 mediates the elimination of cells by PCD in higher plants. So what happens in aging seeds? Before we go into the next slides, I just want to show this slide which looks at the response of Arabidopsis seeds to irradiation. So in this slide, we've taken a Arabidopsis seed and it's been irradiated as previously at 0 hours imbibition, but after 48 hours of stratification. And then we look at the incidence of programmed cell death during the subsequent progression of germination. So we can visualize the appearance of programmed cell death as the appearance of dead red cells. And actually, we do not see the incidence of programmed cell death until the completion of germination with the emergence of the radical through the seed coat. And the middle panels are markers for cell cycle stage. So this slide illustrates an important point that even though the seed is subject to DNA damage very early in imbibition, we only start to see the induction of programmed cell death after the completion of germination, and it is coincident with the onset of cell cycle activation shown by EDU labeled nuclei here. These are cells entering DNA replication or S phase, and we can clearly see that therefore DNA damage incurred in seeds induces low levels of PCD than found in seedlings, but that this PCD is only detectable after the commencement of cell cycle and the completion of germination. So what happens in aging seed? So In this slide, we're looking at the induction of PCD after seed aging in wild type and SOG1 mutant lines. So on the left, we're looking at the analyses of unaged seed lots. We see representative images of the wild type and SOG1 mutants. We see little incidence of PCD in the germinating roots of unaged seeds. However, when we look at the germinating roots of seeds that have been aged using accelerated aging, you can see here on the top panel, we can see the germination performance of the seed lot. We've got around a 8% viability of the SOG1 mutants, but around a 60% viability of the wild type lines. We can see that the wild type aged seeds is showing PCD, but the SOG1 mutants are not. So another important point is that we see PCD in aging seeds, but that the number of cells undergoing cell death is never comparable to that which we see in seedlings exposed to DNA damage. And also, we do not see PCD in all germinating seeds or seedlings. So in the next slide, we've quantitated the levels of PCD in a number of age repair mutants. So as you've previously seen during the course of my talk, we always use the same standard aging conditions and use the 7-day aged seed as a seed which has lost vigor but not viability, but 14 days aging results in significant loss of wild type seed lot viability. In this panel, we've presented representative images on the left and graphical quantification on the right. As I've just mentioned, we do not see PCD in all germinating seeds after aging. So we've actually scored the number of roots in which we see PCD. So in the unaged seed, we see the incidence of PCD after 7 days aging, that is around loss of germination vigor, but not viability. We see around 30% of seeds will display some PCD. This is much lower in SOG1, ATM, and ATR, indicating that in seed aging, DNA damage induced by seed aging is that causes PCD mediated by the DNA damage response in SOG1. However, when we look at the levels of PCD in seeds which are deficient in repair of double-strand breaks, as in our mutants deficient in non-homologous end joining, here are double DNA ligase 4/6 mutants, we identify that even unaged seeds show some incidence of PCD. So around 60% of unaged DNA ligase 4/6 mutant seeds carry cells undergoing PCD, and this increases to nearly 100% in seeds that have been aged to lose germination vigor, but not loss of viability. So seed aging is associated with striking levels of genome damage. So does DNA damage incurred in the aging seed impact on genome stability and growth of the future plant? So to date, it's not really understood how seed vigor can impact on seedling, how seed vigor impacts on seedling vigor at the molecular level. So in this slide, we're using a transgenic reporter line in which the appearance of a blue sector represents the incidence of mutation. So here we are using reporter lines of Arabidopsis in which a mutation in a G16 microsatellite, that is a run of 16 Gs, can lead to Gus reporter expression and hence the appearance of a blue sector. So basically, the reporter gene is out of frame, and when a mutation occurs, we can visualize this as the appearance of the blue sector. So we wanted to look at the impact of seed aging on mutations in the subsequent seedling. So on the left, we look at the incidence of mutations counted by Gus spots per leaf, and the representative image of the first true leaf germinated from unaged and progressively aged seeds. So we identified, as shown both by image and by quantification, that the true leaf germinated from unaged seeds carries a low level of frameshift mutations. However, as seed aging increases to 7 days, that is a point at which we've lost vigor but not seedlot viability, we start to see a threefold increase in mutations. And this increases as seedlots continuously age and we start to see loss of seed viability. However, the take-home message from this slide is that even small changes in seed vigor, so for example, seeds developing on the mother plant under suboptimal climate conditions, may result in increased mutations in the seedling. And here you see some of the results with a reporter line in which deletion by recombination leads to reconstitution of the Gus reporter line. In other words, mutation is causing reconstitution of the Gus reporter gene and hence expression in the leaf. This is a reporter for recombination, and we see that in the 7-day-old, or the 7 days seed aging, which does not result in seed vigor, leads to a 5-fold increase in recombination frequency. We subsequently looked at the effects of DNA repair deficiency on seedling vigor. So in this slide, we are using mutants deficient in the gene Q80, which is involved in double-strand break repair in Arabidopsis. We identified that the germination of these mutants after aging is the germination performance of the mutant Q80 seeds is hypersensitive to aging compared to wild type. We had then looked at the fresh weight of the seedlings germinated from these unaged and aged seeds. Here, shown in the Q80 data is shown in grey. We identified that after seed aging, we see significant differences in the biomass of the seedlings obtained from the aged seeds, so around 10 milligrams per seedling for the Q80 mutant and around 20 milligrams for the wild type. We then looked for the molecular basis of this difference, and we looked at levels of programmed cell death in the unaged and aged wild type and Kuwaiti lines. Very briefly, we see PCD is initiated in the aged seeds around 4 days post stratification and persists for a number of days. On the right, we've presented the data as surface area of cells undergoing PCD. So we can see that PCD is occurring in seedlings germinated from aged seeds, especially the Kuwaiti mutant seeds, which suggests that elevated PCD in seedlings is a molecular link between seed vigor and seedling vigor. So this summarizes the last section of my talk. We have presented evidence that in the unaged seed, the maintenance of the genome leads to the intact genome and a healthy seedling. However, after seed aging, we identify genome damage is associated with an elevated level of mutagenesis and genome instability in the germinated seedlings, and also that PCD is identified in seedlings from the seedlings germinated from aging seed, which may contribute to reduced seedling growth. So finally, I'd just like to conclude, to summarize the findings I presented today, that DNA repair factors are important determinants of seed longevity, that resistance of Arabidopsis seeds to DNA damage is lost in germination, coincidence with cell cycle activation, that key developmental and transcriptional differences in the seed DNA damage response promote germination and establishment. And finally, that genome damage accumulated in seed aging impacts on seedling vigor, plant growth, and genome stability. And finally, I'd like to thank the group leader, Chris West, and also the majority of this work was done by myself and Chris, but there's also been input from Rosalind Latham and two master students, Mona Al-Sharif and Ithia Balobad. I'd like to thank our UK-based collaborators for the work I've presented today, Darpeng Wang, now at Imperial College, Cliff Bray at Manchester, and Bill Finn-Savage and Steve Footit for collaboration on the ATM work. Okay, so thank you for your time.
Thank you very much. Very nice talk and very insightful in the topic of cell cycle. It kind of recalled me my biology lessons. I'm not in the cell cycle of in plant cells, but I still remember my cell cycle courses from animal science. So it kind of reminded me. So do you have any questions in the audience? Alma has a question. You should probably activate your microphone.
Thank you, Wanda, for this wonderful talk. Can you hear me?
Yes, I can. Thank you.
So I have a curiosity because I know that in the DDR signaling pathways, there are also some putative.
I didn't hear the last part. Sorry.
No.
Okay, can you hear me?
Yes.
Oh, sorry, there is a problem with the microphone. Just a curiosity, because I know that there are also some SOG1 independent pathways acting in the DDR context. Can you comment a little bit about the roles or what we know about these SOG1 independent pathways?
Well, I think, okay.
Alma, silence your microphone.
Well, I think that an important point is that SOG1 controls a lot of the plant DNA damage response, but not all, and there's still an awful lot that we need to learn about the plant DNA damage response. For example, in other eukaryotes, ATM responds to oxidative stress, and there is some evidence that that the DNA damage response is involved in oxidative stress responses in plants, and also even things like pathogen response, but that's not very well characterized. So, and also with respect to SOG1 independent pathways, yes, I'm afraid I don't know very much about SOG1 independent pathways in control of plant programmed cell death. In fact, plant programmed cell death and plant programmed cell death specifically in response to DNA damage remains quite uncharacterized at the molecular level, because of the DNA damage response, it's the part of the DNA damage response which is most different from mammals and yeast. So it's known that SOG1 controls the plant DNA damage response, but a lot of the downstream players still remain to be identified. So, I guess my response to your so I guess it's not a very specific response to your question, but I guess it is that there are many different pathways in the DNA damage response, and also lots of different repair pathways and factors operative in higher plants. So that yes, I think that although a lot of focus is put on the known players in the plant DNA damage response, there's still lots we don't know, and it's just much harder to identify novel components of the plant DNA damage response. Also, SOG1 was itself identified in a forward genetic screen. So you know, it takes so much more effort to work or to identify, you know, truly novel players that are just specific to plants. And lastly, I'd also point out that so a lot of my work is with Arabidopsis, and obviously we have very powerful resources with mutants, but one question is that if we use mutants to study DNA repair pathways, but obviously one of the things you do find in plants is that you can knock out lots of pathways and the plant will still remain viable. A situation you don't really get with yeast and mammals. So what actually happens is that there are many different double-strand break repair pathways and factors. There are backup pathways for non-homologous end joining. For example, lots of different pathways for homologous recombination, single-strand annealing. But what happens in a plant cell might be different from a mutant plant cell, because that's not a natural situation. So I think that you have to take that into account as well, that when you're looking at mutant analysis data that indicates that a pathway is important, I guess you have to ask the question, what is actually going on in the plant really? So for example, we can knock out the main non-homologous end joining pathways, and then other pathways, for example, you can knock out ligase 4, you can knock out DNA ligase 6, and DNA ligase 1 will mediate non-homologous end joining. But
Thank you so much.
Very much. As you were speaking, as I said, I was also looking in because I'm not familiar with all those genes. I found that the SOG1 is similar to P53 in mammals, in animals. So kind of is related to the arrest of a G1 before the restriction point, right?
yes, but it's often proposed to have a function similar to P53, but it's unique to plants, and PCD is quite distinct from apoptosis, and even in the world of plants, there are many different types of programmed cell death, or that programmed cell death occurs in response to many different stimuli. It can also be a natural part of development. So specifically, SOG1 is involved in PCD in response to DNA damage. But as we were in response to Alma's question earlier, there's lots we don't know. And it's very different. I mean, we've tried to find downstream factors in plant programmed cell death downstream of SOG1, but it's very difficult.
Yes, I think it's very interesting. And just you mentioned that all your work. Sorry, you don't see me. I'm in the hall. All your work, you've been working with Arabidopsis, which has a relatively small genome.
Yes.
What do you think this DDR response when you face like plants with bigger genomes, either polyploid plants? How would you presume that the response would be like plants will be more resilient, probably, or resilient to this type of damage, or
well, I think that one important point is that the plant DNA damage response and repair factors are very highly conserved across all plants. There has been some work done with maize, ATR, ATM, for example, also in rice. There's been work done on the plant DNA damage response. But maize, for example, has got a large genome, and it does appear that there are slight differences. So I guess but not enough work has been done at this moment in time to really answer the question: are there going to be fundamental differences? I think that with plants with larger genomes, I think that it's going to become even more critical that genome maintenance is maintained. We've done most of our work is with Arabidopsis, but we have done some translational work with different crop species, and so far we found that all the genes that we've looked at are highly conserved, and we've certainly found that the transcriptional DNA damage response is present in quite a wide range of species, indicating that it is an important component of germination in monocots too, for example.
I see. Thank you very much. Any more questions? I'm just checking if there's questions in the box. No, there are no questions. So thank you very much.
Okay, well, thank you for the invitation.
I enjoyed very much your talk. Thank you very much. So let's move to the next talk. Andrea, would you please, Alma, introduce Andrea?
Yes, Andrea Pagano is a postdoc here in Pavia at the Department of Biology and Biotechnology. So the main topic, the main focus of his research activity is again the response to genotoxic stress, the seed repair response, particularly in under seed priming conditions. So he will tell you the results of his research. Andrea, it's yours.
Here. Okay, can you visualize the presentation? No, non vado qui. No. It's okay. Can you see the presentation? Can you hear me? I don't know.
We hear you perfectly, and the presentation also we see it. But you can you make it bigger with the PowerPoint?
Yeah, I can try. I'm not
Full
sure it will work because here we had a bit technical problems. I'm trying. Can you see it properly?
No, it's still the same. Probably it's not working for whatever reason.
Let me try because from here. Okay. I have to close this maybe. No, that's another thing. Okay. Okay. Sorry for the technical issues, but from here we are not used to this. Okay. Let me try.
You cannot swap the slides.
Sorry.
The slides. Can you swap the slides or not?
So just yes, I have. It's open, but I'm trying to share to put it bigger. But it asks.
Try to move the slides.
Yes, no, I'm sharing.
No, that move. I mean, like a page up or a page down, to see if you can change the slides.
We are just calling a second. Sorry, da condividere. Che devo fare?
Condividere la presentazione.
Non lo vedo.
Okay.
Okay, from here, but to put it bigger, I have to do it from there. I'm not sure. Sorry, I miss it. Because ho provato da là, ma non lo vedo.
Can you see it now?
We can see the first page, but we don't know if you can swap the slides.
Ah, yes. You are seeing other slides, or just the first one?
I'm just seeing the first one.
Ah, okay. So there is the same problem as before.
Yes.
Okay.
Now it's moving.
It's moving?
Try again. Page up.
It's moving.
Page up, page down. Just see if you can move the entire presentation or a couple of slides.
Yes, I think it's moving, yes.
Okay.
Yes, but it's also big, larger screen, because I'm not sure it's big, but I don't know if we can achieve both a big screen and moving slides.
Just try one thing. Try to save the presentation as a PDF. If you don't have any animation or something, you can open with the PDF viewer, and probably it's working better.
Yes, just a second. I'm trying to see if I can share the PDF. Let me just try to open it. Okay. Okay, let me try. Okay. Yeah, this, okay.
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 Finestra.
This. Okay, do you see it better from the PDF?
I think now it's better. Maybe you can just put it in full screen.
Yes, I have to, for full screen, I have to go here.
Is it okay?
It's full screen from the PDF or what?
Yes. Yes.
It's okay?
What was the combination, the key combination? Control L maybe? Yeah, you have the yeah, modal, no, I don't know which one.
Can you see it?
Great, perfect. That's perfect.
Sorry, let's check if it goes up and down. It goes up and down now.
It works. Okay.
Excellent, and also the animation.
We made. Okay, thank you for that. Your patience. It's a bit every time. Okay, Andrea Pagano, thank you for this nice occasion to share ideas about seed priming. I'm going to talk about overpriming, an informative system to explore the seed response. So, ironically, is when seed priming is too much. Seed priming encompasses a family of well-established techniques used to enhance germination, to improve germination performances in terms of the percentage, speed, uniformity, and stress tolerance. In general, seed priming protocols rely on an incomplete inhibition that pre-activates seed metabolism, followed by a dryback stage that leaves the seeds in a primed state, so prepared for this improved germination performance and stress response. The working principle is the activation of pre-germinative metabolism. Orthodox seeds are able to withstand important dehydration events, both during maturation, but also during early stages of inhibition, thanks to the endowment in hormoprotectants, the activation of DNA damage response, host scavenging, and especially the glassy state that stabilizes cell structures, and the accumulation of late embryogenesis abundant proteins during seed maturation. All this complex machinery of factors helps orthodox seeds to survive dryback events, also in the context of seed priming. So it is essential for an optimal result during priming. Nonetheless, when inhibition or seed priming, in the context of seed priming, is prolonged and seed reaches germination, then they lose their ability to withstand important dehydration events without consequences on priming efficiency and with worse performances than controls. This condition, this loss of desiccation tolerance in early seedlings, in the context of seed priming protocol, is called overpriming, and it severely compromises the effectiveness of priming. Of course, it's the events that lead to overpriming from a normal, optimal primed state are complex and partially unknown. So they are function not only of water content, but also of time, germination progression, and so the situation is quite complex to be called. In general, it's due to the depletion and of the components that ensure desiccation tolerance, and then in the hydration, the disassembly of the intracellular glassy state that grants desiccation tolerance. Overpriming has consequences on priming efficiency because it is difficult to predict, and so it needs to be optimized, often with empirical approaches, depending on species, cultivars, and even seed lots, because it's quite difficult to predict a priori when an overpriming state will occur, and subsequently what is the optimal inhibition time for each seed lot. This has economical implications, but represents also an interesting experimental system to study the loss of desiccation tolerance between seeds and early seedlings. That is a nuanced area of seed between seed pregerminative and germinative metabolism that is interesting for this reason. Of course, overpriming seeds has a much worse performance in the field and even in our experimental systems in vitro. And so we first try to identify contrastive phenotypes in this sense. As a general aim of the research lines in which I was involved concerning overpriming, the general aim was obtaining a model of post-priming signification response as detailed as possible, considering different levels, starting from the indeed the biometrical parameters that define the occurrence or non-occurrence of priming and overpriming conditions. Biometrical parameters we typically evaluate is germination performance through several germination parameters, and seedling morphology, seedling phenotype, and so root and shoot length, relative water content, biomass, and so on. Then other elements to decode the response to priming and overpriming include the DNA damage profiling and DNA damage response. Also the oxidation and antioxidant response, and the transcriptomic dynamics involved in this kind of response, and the chromatin remodeling influencing and regulating transcriptome. And of course, we tried also to retrieve metabolic signatures that can be accumulation of hormones, antioxidant species, antioxidant molecules that can be indicative of the processes that are activated or inactivated, and possibly acting as a hallmarks for seed quality, so seed vigor, seed quality, and efficiency of seed priming, or the occurrence of overpriming. Hopefully they can be implemented in seed quality assessment protocols, so for an early diagnosis of seed quality traits that think of the presentation of the animal. For example, if some metabolite can be linked to volatile compounds, it can be useful to monitor these events as a continuation link with that other experimental system. Otherwise, the metabolites that can be relevant for antioxidant response or DNA integrity can be useful to complement as a priming agent to complement the seed priming protocols in the inhibition state to protect the primate seeds from the effects of overpriming or for other stresses that can occur in early germination. So the possibilities in terms of seed quality assessment and improvement are many. To transpose these ideas in terms of experimental system, we decided to use Medicagotum catula as a first model organism in this study, and Medicago sativa for proofs of concept of certain levels of analysis we have adopted. As a model Pavia. The established experimental system to compare the primed and overprimed seeds consisted of a prolonged inhibition, 24 hours, that for Medicagotum catula is quite long, at the end of which two subpopulations were configured, primed and unprimed. They were easy to distinguish Medicagotum catula because simply primed seeds do not display radical protrusion, while overprimed seeds display a radical protrusion in the order of 1 or 2 millimeters. Of course, we had hints from previous studies and from the literature establishing priming and overpriming criteria in terms of radical protrusion. So it was an interesting system also because it was the occurrence of priming and overpriming can be spotted at the end of prolonged priming. As expected, we characterized the system, and overprimed seeds developed into aberrant seedlings with severely impaired radical growth, as opposed to seedlings, the normal seedlings developing from primed seeds. So this was the morphological and phenotypic criteria to distinguish priming and overprimed seeds. From a previous study, we highlighted that the tissue that is more exposed to the effects of overpriming, so it is more exposed to
post-priming dehydration is the hypocotyl radical axis. This can also be linked to the fact that the aberrant seedlings developing from overpriming display a strongly impaired radical protrusion. Subsequently, this tissue, hypocotyl radical axis, was chosen for molecular analysis and for the sampling for molecular analysis. First of all, this time on all seeds, we quantified the accumulation of reactive oxygen species through the chlorofluorescent diacetate assay during the dry back of primed and overprimed seeds. As you see, overprimed seeds displayed a much higher ROS emission, ROS levels during dry back compared to primed seeds. And again, they were localized in the radical tip, as shown by the histochemical stain with DAB, in which the positivity to hydrogen peroxide was mostly localized in radical tips. We quantified the expression of genes involved into antioxidant response, highlighting overexpression in overprimed seeds during dry back. So highlighting also a response to ROS accumulation. We passed to the level of DNA damage in response to priming and overpriming, and again, the overprimed seeds during that seeds, but with early seedlings, very early seedlings, displayed higher levels of DNA damage, single and double strand breaks, compared to primed seeds during dry back. This assay is alkaline single cell gel electrophoresis, the comment assay. At the same way as the ROS, we highlighted upregulation of genes involved into DNA repair to DDR, again in overprimed seeds during dry back, consistently with the higher levels of damage observed in this during dry back of overprimed seeds. We considered also a hallmark of DNA damage at the level of the chromatin, that is the histone variant gamma H2AX, that is present when the strand breaks occur as a first signaling for the recruitment of DNA repair machinery. This immunolocalization in the nucleus of primed and overprimed seeds highlighted accumulation of an higher density of gamma H2AX foci in overprimed seeds toward the end of the dry back step. For this analysis, I thank Professor Vicent and Dr. Zannino who helped us with this analysis and electron microscopy. I thank them also for their help in studying the nucleolar dynamics in response to priming, overpriming combined with dry back, because it highlighted the possible involvement of nucleolar dynamics in the response to priming and overpriming. Indeed, the nucleoli of primed seeds were morphologically different than the nucleoli of overprimed seeds that displayed a tripartite organization of the nucleolar chromatin compared with the vacuolization observed in primed seeds. This is indicative of a different transcriptional activity status that distinguishes the two nuclear morphologies. After dry back, the nuclear structures consistently with water loss collapsed in both treatment groups. We consider this aspect of nucleoli because of previous hints from previous studies highlighting possible roles of the nucleolus as in coordinating stress response. Consistently, we quantified the expression of the precursors and mature forms of ribosomal RNA and also genes involved in RNA processing, highlighting overexpression in overpriming seeds during dieback, and a trend that was confirmed in medical causativa, and that corroborates the putative involvement of the nucleolus in the response to overpriming. Another aspect of stress response that we considered in this experimental system was the involvement of the nucleolus plastid retrograde signaling, especially concerning this signal molecule, the 3-phosphadenosine 5-phosphate, that in Arabidopsis has been linked to roles in development and stress response. The levels of PAP were much lower in overprimate seeds and much higher in primate seeds, along with an upregulation of a gene involved in PAP turnover. To summarize the result of this from this experimental system, we conclude that we started from the biometrical definition of the occurrence of priming and overpriming, defining when respectively desiccation maintenance of desiccation tolerance and loss of desiccation tolerance during post-priming dieback. Different putative hallmarks of the stress response to post-priming dieback in priming and overpriming were defined. So, like the decrease of PAP with overpriming, and the increase with overpriming during dieback of the levels of reactive oxygen species, expression of antioxidant genes, and accumulation of DNA strand breaks, and markers of DNA damage at the level of the chromatin in the form of this histone variant, and the overexpression of genes involved in DNA damage response, along with an upregulation of mature forms and precursors of ribosomal RNA. As a further development of this research line, we are currently analyzing further data sets concerning the chromatin dynamics associated to priming, overpriming, and post-priming dieback. And we are planning to validate these results on a wider panel of legumes, both model legumes and neglected legume species. Concerning the first development, it is being carried out on a simplified experimental system consisting of primate versus overprimate seeds before and after dieback. It is being performed with the support of Società Italiana, so the Italian Society of Plant Biology, thanks to a grant by the society, and with the collaboration of colleagues at the University of Paris-Saclay, that I thank now, Professor Penament and Renaud. This further development of the primate versus overprimate research line will integrate RNA-seq analysis, so highlighting the transcriptomic dynamics in response to priming, overpriming, and dieback, and the changes in chromatin accessibility to highlight the regulatory networks integrating the chromatin condensation and decondensation with the transcriptomic changes occurring in response to overpriming and post-priming dieback. We are also considering the level of hormonal regulation, especially the accumulated turnover of abscisic acid, because it's one of the key phytohormones regulating both germination and stress response in seeds, and the accumulation of polyamines as a possible antioxidant compounds in the context of post-priming dieback. Also because with gene expression analysis, we highlighted the differential expression of spermidine synthase and spermidine synthase. These analyses are being performed with the help of Professor Daniele Dondi and his laboratory. For the opportunity to present here and for the working environment and collaboration, I want to thank my colleagues of the Seed Molecular Physiology Lab and Plant Biotechnology Lab, my supervisors, and our national and international collaborators. And also thank you for your attention. I hope everything was clear. If you have questions, I'm here.
Very much. Very nice and insightful presentation, very complete. Let me check first if there are questions in the comment box. No, I think it's too late for the observers at the other side of the line. But we have a question from Wanda Waterworth. Please, go ahead.
Yeah, it was a really nice talk. I was just curious, do you think that overpriming and the loss of desiccation tolerance are associated with the initiation of the cell cycle in germinating seeds? Because and also that the increasing DNA damage you get with overpriming might, at least in part, arise from cell cycle starting.
A second. Can you hear me? Yes. Okay, concerning the question about cell cycle, in this research line right now, in these results, we have not considered that aspect. Nonetheless, in the development, so in the transcriptomic analysis we are currently analyzing, and in the chromatin remodeling aspect, we are one of the process we are considering is cell cycle to highlight possible links in that direction, along with other dynamics of DDR and chromatin remodeling. So we hope to corroborate these results with the new results of transcriptomic and looking to provide a more complete vision of both in terms of cell cycle and activation of DNA damage response. Because for this study it was QRT-PCR, and now we have modules, studying modules of gene expression specifically for cell cycle, ribosomal dynamics, and activation of DNA damage response genes and antioxidants. So we hope in the future to have a more complete vision and to corroborate this upregulation of DDR genes in overpriming seeds. Also, maybe not specifically considering, but also the transcription should shut down with dehydration. So it's an aspect also that we are aiming to comment in the future work. So I hope I replied to the question.
Yeah, no.
Basically, I thought that we will know better later. So.
Yeah, no, it's just there are a number of tables which kind of indicate there might be a link.
Sorry. The microphone is giving us too much feedback. More questions? I'll have more like from my side, more as a comment rather than a question. So we're working on plant hormone regulation of abiotic stress responses, and of course ABA is always there. So during the germination, you mentioned you want to explore the role of ABA turnover from a reversible inactive form to the active form. Have you investigated what happens to ABA during the imbibition and then the drive back in those overprime seeds?
We are. Can you hear me? Can you hear me?
Can hear you.
Yes. Yes. Okay. Currently, we are quantifying what's happening during dehydration. During germination, theoretically should decrease. But besides the quantifying cystic acid, we are considering also sensitivity and expressions of genes codifying from sensors and from the sensitivity to abscisic acid, because there are some works highlighting that it's not only about abscisic acid levels, but is especially abscisic acid sensitivity in several instances to regulate the response to abscisic acid rather than only ABA levels. So right now we are quantifying abscisic acid, and hopefully we will be able also to quantify abscisic acid glucose ester to have an idea of the turnover. But at the same time, we are hopeful also to find even more promising data on the side of ABA sensitivity. So by studying the expressions of genes that can be linked to ABA sensitivity. And of course, another module we are studying in RNA-seq analysis is abscisic acid biosynthesis, catabolism, and sensitivity. So we are going toward quantitation with HPLC, but I'm even more optimistic to have information from the side of RNA-seq analysis. So yes, we are considering this aspect.
Thank you very much. Just to close this circle, it has been long discussed the relationship between ABA with ROS accumulation, ROS production. Several enzymes, for instance, these respiratory burst oxidases found in membranes. Some isoforms seem to be induced and regulated by ABA. I'm wondering, do you think the ROS we observe during this germination response comes from these enzymes? Do you think there's any relationship? What's your hypothesis? Is there any relationship with ABA, or what do you think these reactive oxygen species come from?
Indeed, the link between abscisic acid dynamics and polyamine accumulation and ROS levels and antioxidant response was the aim of one of the main aim of my research project, one of my postdoc research projects. So yes, we aimed to investigate on this direction. From the side of ROS accumulation, we hypothesize in that context those levels of ROS can be considered as proxies of stress in this specific context, and so probably leakage from damaged organelles, probably the cellular origin is there. Concerning the relation between abscisic acid and antioxidant stress response, we aim also in investigating on this side to see if ABA is inducing an antioxidant response in this context. So not yet, but with the general aim of characterizing what is the role of abscisic acid in this context. we are moving both with upper quantitation and RNA-seq analysis to try to fill also this gap on what is the link between antioxidant ABA and containment of ROS levels in the context of post-priming dieback. Again, we are considering also differentiated tissues. So in the radical tip, this is particularly evident. So we plan to go also in this direction. So I'm not sure if I replied properly.
Yes, thank you very much. I don't know if there are, could you please silence the microphone? No, thank you. Any more questions? Okay, thank you very much for your nice talk. I think we can move for the last wrap-up talk with Alma. I will open the file. So I've been gathering all your talks, and so I prepared kind of a wrap-up talk here. Can you see it? Now you can see it. Now, can you see it?
Yes.
Okay, great. So I think probably with the YouTube, it takes some time to refresh. Well, this is kind of like a closure session. We'll just mention those things we learned, because we learned a lot. I learned a lot during these different talks, and I expect that this is the beginning of a lot of interactions and potentially collaborations between the different participants in this workshop. I don't know, you want to begin? I can begin, whatever you want. Alma.
As you prefer. Start and I will follow you.
I will just begin at more or less at the middle. I will stop. Okay. Full screen here, and I will be moving the. Okay, so we have entitled this last wrap-up presentation like output, expectation, and opportunities of seed priming, what we consider is a green tool for sustainable agriculture. And yes, all we have been talking during this workshop is intimately related to the project objectives of BENEFIT-Med and Optimus PRIME. With the first talk, we learned the importance on knowing exactly what defines the viability of seeds, which mechanisms control seed viability and longevity, thanks to the work of Dr. Eduardo Bueso. Then we had the chance to listen to Adriano Grifo and their high throughput methodologies that he worked to set up and put in motion to measure the production of different reactive oxygen species and nitrogen oxygen species as markers of seed viability and aging. Later, we had the pleasure to enjoy the talk of Dr. Lloyd Graju. He showed his amazing work on different seed priming strategies, how this is linked to transcript profiling and different expression of genes and accumulation of metabolites, and how this contributes to the improved behavior of the germinated plant. And we also had the participation of Dr. Beatriz Julián from INAM here in Universitat Jaume I, who explained the different opportunities on biocompatible materials, new materials that could be used as delivery materials to deliver the different priming treatments to plants in seeds or even in planta, and also contributed to a more sustainable agriculture. I will leave the floor to Dr. Alma Balestrazzi.
Thank you. So, with the talk by Cinzia, Bertea, we went even more in depth into the issues of plant biostimulants, and so what is their role, their essential role in promoting the circular bioeconomy, so the value from this point of view, while using agro-waste materials or other to produce new technology, and the relevance, the importance to go more in depth at the molecular and cellular level to gain insights into the molecular players that control the response to biostimulants, also in view of this variability, heterogeneous response that often is observed when we manage different species, genotypes, and even seed lots. And then there was Patricia. Patricia again, the topic of biostimulants. So in this case, what was really very, very intriguing to me is the systematic development of a smart, let's say, screening method, because we must also consider the fact that we need to speed up this essential step. So speed up the screening of many potential candidates as biostimulants. And then what is much closer to my research topic, the talk by Wanda. And so this reminds me what is a fascinating issue, because seeds, if they want to preserve genome integrity and they want to repair DNA, they must stop cell cycle. So the seed either repairs its DNA damage, or either it grows. So that's quite an intriguing issues, and I'm sure that there will be a lot of further future development on this issue. And then again, the talk by Andrea. In this case, Andrea is exploiting a drawback of seed priming, something that we don't want, or at least the operators, the seed technologies, the industries, they don't want. But this drawback turned out to be really very, very informative. So that's, I would like to underline that we need also creativity and flexibility in, looking for new systems, new experimental systems. And of course, all the talks have been fantastic, really, really fantastic. So one full day about seed priming, science, technology, different aspects. Well, that's wonderful, for me at least.
Yeah, I am the same opinion. All the talks have been really interesting. I personally learned a lot, and also I found very interesting aspects to investigate in our own research. Unfortunately, we didn't have the opportunity to show any of our research, but I'm sure we'll have the chance in the next in our next workshop to see some preliminary results of our research. Thank you very much for this opportunity. It was a pleasure to have you all here, and I really thank for your availability, for your kindness, and for the patience. This was the first time we organized such a hybrid, two-sided, digital, virtual, face-to-face, and many more things workshop. And of course, we had different problems, but I think we volunteer to solve these problems and to make them work, make this work. So thanks again. and hope to see you soon. Thanks a lot, Alma. I don't want you want to say something.
Thanks to the Spanish team, to the Optimus Prime team from the side of the BENEFIT-Med team. So I'm sure we're going to work again together, because that was very, very nice experience. And so thank you to all the speakers for being with us today.
Thank you very much to you for inviting me. Thank you very much. It was really interesting day.
Okay, and all the people that it's watching this broadcasted workshop, thank you very much for your presence, for your interest in this type of emissions that usually we are not artists, we are not musicians, we are not TikTokers, but still we are able to draw some attention from the general audience. So I'm glad on this. So to all of you, have a nice evening and hope to see you soon.
Bye, thank you.
Bye, thank you very much.
Bye, thank you very much.